Diabetes, a disease that has afflicted humanity for thousands of years and that persists worldwide to this day. According to United Nations data, every 5 seconds, someone in the world is diagnosed with diabetes. The number of people who suffer from the disease has already surpassed 500 million and continues to grow. Type 2 diabetes accounts for the majority of cases. Insufficient insulin secretion from pancreatic beta cells and the body's inability to respond to insulin properly cause blood sugar levels to spike. This can lead to complications in the eyes, feet, and kidneys, and in severe cases may be life-threatening. Although the pancreas has been known to be the key organ in regulating blood sugar, throughout medical history, diabetes treatment has yet to see a breakthrough. The human body itself has various physiological mechanisms that, once found and harnessed, could exceed all expectations. In the early 20th century, the concept of incretins was taking shape. Scientists realized the intestines may secrete hormones that stimulate insulin release from the pancreas, but their nature and functions remained a mystery.
But what was observed many years ago was that when you take glucose by mouth, you get more insulin release than if you inject the same amount of glucose right into the vein. So the insulin stimulation is enhanced by something coming from the fact that the glucose went through the intestine. And so these hormones were called incretins. So the question was, what were these hormones?
Only around the 1960s did the mystery begin to unfold. The first incretin to be identified was GIP, gastric inhibitory peptide, which is secreted by K cells in the small intestine. It suppresses gastric acid and is glucose dependent. It induces insulin secretion as blood sugar rises. However, scientists found its effects limited, which meant other incretins had yet to be discovered. The 2024 Tang Prize in Biopharmaceutical Science was awarded to Joel Habener, Svetlana Mojsov, and Juul Holst. These three scientists decoded the decades-long mystery of incretins and drove massive advances in diabetes and obesity treatments. The story began many years ago. Professor Habener, a towering figure in endocrinology, graduated from the UCLA School of Medicine. When the Vietnam War broke out, he was drafted, not to the battlefield, but to the National Institutes of Health, where he did cutting-edge DNA research.
We had a lot of seminars on the field activities that are going on in molecular biology, which is the new name for the biology of the DNA and RNA and all. So it was a very rewarding experience.
He then joined Mass General Hospital's endocrinology division, while also collaborating with MIT's thyroid research group. With funding from the Howard Hughes Medical Institute, he was able to start a new project. Habener chose to focus on glucagon research.
So I switched from parathyroid hormone to glucagon, and nothing much was known about how glucagon is made in cells.
Glucagon, secreted by pancreatic alpha cells, breaks down glycogen, raising blood sugar levels when glucose levels drop too low. It works counter to insulin, another key hormone in blood sugar regulation, and it was then that emerged a breakthrough, a new cDNA technology that allowed scientists to study gene expression across species, which are often hard to pin down. Through amplification and recombination, the technique facilitates gene function testing and protein analysis. Habener thought of utilizing the technology for experiments in rats, but hit an obstacle.
The research committee said that we could not go forward with the recombinant DNA studies in warm-blooded animals because the moratorium had been declared. That's a temporary halting of research using recombinant DNA. So we got the idea arose that we could use maybe a cold-blooded animal.
To keep the project alive, a graduate student suggested switching to anglerfish, which were common in nearby Boston Harbor, as well as ignored by locals. What is remarkable are the results of that choice.
And the other was that we didn't realize at the beginning, I didn't realize, I should say, that the Brockman bodies of the anglerfish contain the endocrine cells, and it's a separate organ from the exocrine pancreas. The enzymes that would, you don't want because they degrade proteins and RNAs. So we would get the endocrine cells in a body about the size of a big marble. And they were very highly enriched in messenger RNAs for glucagon, somatostatin, and insulin, and all the hormones of the pancreatic islet snake.
Fate smiled on Habener's team back in the early 1980s when cloning the preproglucagon gene from anglerfish. They were surprised to find it contained another peptide sequence.
Another sequence, which was very much a mystery and a surprise, and it was not glucagon, but it was very close to glucagon. We called it glucagon-related peptide, and actually resembled more closely GIP, which is gastric inhibitory peptide.
Peptides are short chains of amino acids, almost like smaller versions of proteins. Habener thought the peptide they discovered might be linked to the incretins they hoped to find. Once restrictions on warm-blooded animals were lifted, the team resumed rat experiments. They proved that this highly conserved gene existed in mammals and identified the glucagon-like peptide 1, GLP-1, as very similar to GRP in anglerfish.
The proglucagon, we called it, contained a sequence of not only glucagon and GLP-1, but a GLP-2, and actually had also, before we finished the rat studies, Graham Bell's group at Chiron had determined in the hamster that the cDNA had the same structure that we were finding.
The role of GLP-1 was identified by Professor Mojsov. Born in the former Yugoslavia, now the Republic of Serbia, Mojsov developed an interest in observing nature early on.
And when I was a child, that was manifested in my interest in learning about the world of butterflies and insects. And that was very easily accessible to me. My family lived close to a big park, and I could just easily stroll in the springs and summer and observe the activities of the butterflies and insects.
While visiting a museum of natural history, she realized these animals could be observed up close, and so she began collecting them.
I had to develop a skill in order to preserve them without destroying them. And thinking back now, those were my first experiments.
It was in high school that she discovered her love of chemistry. At the University of Belgrade, she studied physical chemistry. The university had excellent courses and emphasized lab work. It also encouraged students to make use of campus facilities.
I believe it was the overall experience which I had during the undergraduate at the University of Belgrade in the Department of Physical Chemistry that made me decide that I want to be a scientist.
After graduation, Mojsov was encouraged to study overseas. She chose Rockefeller University in New York and studied under the renowned Bruce Merrifield. Professor Merrifield invented solid phase synthesis to easily assemble amino acids into peptides, for which he was awarded the Nobel Prize in Chemistry in 1984.
You can think of it as assembling a glass bead necklace. You put one glass bead at a time, and each colored glass bead, and each color represents a different amino acid.
As a graduate student, Mojsov developed an interest in glucagon and produced it using solid phase synthesis. After graduation, she stayed at Rockefeller as a researcher, and with Merrifield, she co-authored major papers on glucagon. She later noticed research done on anglerfish. In 1983, she became head of the Peptide Synthesis Core Facility at MGH, when Bell's team just published the hamster preproglucagon sequence, which contained over 160 amino acids including various peptides. Drawing on her experience with glucagon, right when she saw the 37 amino acid sequence of GLP-1, she made a bold hypothesis.
I realized immediately that there was an arginine residue at position 6. And if one is to cleave that arginine residues, there will be a shorter peptide of 30 amino acids, which I called 7 to 37. It was just a generic. And I predicted that that's going to be the active sequence.
She explained that comparing glucagon against the amino acids between the 7th and the 37th positions of the GLP-1 peptide would reveal the truth.
If this is GLP-1, which was predicted by Bell, 37 amino acids, and if you align it with glucagon like this, they're kind of not completely aligned with each other. But if this arginine residue here at position 6 is cleaved by enzymes, then it moves here. And suddenly you have this perfect fit. And that's what made me realize that it's going to be the biologically active peptide.
So, to confirm her hypothesis, she used solid phase synthesis to mass produce GLP-1 7 to 37.
But the whole idea at the beginning was that I will make enough material so that I can use it in the experiments to develop methods to show that it exists in the intestines, and then use it also in that to show that stimulates insulin release and in clinical studies.
For this, Merrifield sent her a shaker. Solid phase synthesis with shaking and filtration greatly reduces processing time compared to past stirring methods. It took Mojsov several more months to identify a sensitive antibody and confirm peptide activity. Habener's team then invited her on to collaborate. Then, in 1986, they published their landmark paper on GLP-1 7 to 37. Their key findings advanced the understanding of peptides and showed that while the intestines and pancreas express the same pre-pro-glucagon gene, the body's ingenious modifications make it so GLP-1 is released only in the intestine and glucagon secreted only by the pancreas.
It's two different organs. The proglucagon, the same proglucagon, generates entirely different spectra of the hormones.
The two have opposing effects. Glucagon raises blood sugar levels. GLP-1 triggers insulin release to lower the levels. They then injected synthetic GLP-1 7 to 37 into the pancreases of rats and confirmed that it stimulates insulin secretion. In 1987, they published their second landmark paper, highlighting GLP-1 7 to 37's massive potential in treating diabetes. Around that time, at the University of Copenhagen, Professor Holst also identified GLP-1. Holst was curious as a child. He loved music and astronomy. During the 1960s, he was an active student engaged in many activities. He eventually went into medicine, starting with anesthesiology, from which he earned valuable work experience.
I was very fortunate to be working in Israel in 1967, immediately after the Six-Day War, and I was working at the Hadassah Hospital, and it was a great inspiration for me.
Back home, Holst worked at Bispebjerg Hospital in Copenhagen as an assistant anesthesiologist. After getting full certification, he took part in a 14-day surgery where he found he was well suited to become a surgeon.
I loved it, and I loved the drama, I loved the atmosphere, I loved the nurses, and so I liked it, the atmosphere, everything.
Holst spent 16 years as a doctor in the ER. He excelled and moved up the ranks. While he had an interest in intestinal surgery, he never gave up on scientific research. Then, when insulin studies were very popular, a University of Copenhagen professor invited him on as a research assistant.
This was a great success. This was a wonderful time of my life. I was really excited. So I got this position and I got into research, and I was doing more and more research. And The research field that I was in was this new field of gut hormones, hormones from the gut. And that's what I've been doing ever since.
It was precisely this background that helped Holst see how patients' blood sugar dropping after GI surgeries might be connected with GI hormones.
And so this became an interest of mine, you know, trying to find out why was that. And this was linked to the main theme of our work with the hormones, because we were actually looking for hormones from the gut that would stimulate insulin secretion. This is what is called incretin function. So the incretin function was the big thing.
After GIP was first identified, Holst began his own research. He observed something interesting. Certain GI cells reacted to anti-glucagon antibodies. Using his radioimmunoassay expertise, he found hormones in the intestines that differed from pancreatic glucagon, a discovery that took years to explain.
Numerous publications from the time narrowing in what it was, because eventually I found out that it was the glucagon peptide was buried inside this peptide that was from the gut somehow.
The Bell team's findings later confirmed what Holst had identified. It was the first half of the pro-glucagon gene, while the second half contained GLP-1 and GLP-2. This blew the doors wide open for his research.
Because, you know, one thing is a nucleotide sequence that doesn't tell you anything about what is really existing, what the real peptide is, and what the physiology, nothing.
Through experiments, he found that the pancreas has GLP-1 and GLP-2 sequences as well, though they are not secreted there.
And we could demonstrate that GLP-1 and GLP-2 were released from these, from the gut, but not from the pancreas. It was only the big one. So this was, this was an important step. Yes, it is.
He tried injecting GLP-1 into pig pancreases, but saw no results. He later found a workable method.
We used the assays to look for peptides that were looking like GLP-1 in the gut extracts. And we found one, and we purified it and put it on the pancreas, and then now here was a peptide that really could produce insulin, a lot of insulin.
He sequenced the peptide gene and made a breakthrough discovery.
And found out that it was a different peptide from what was predicted. It was truncated by 6 amino acids and had a different C-terminus. But so now we have the structure, and that is GLP-1 as we know it today.
In 1986, he published his discovery of GLP-1. Holst differed from Mojsov in using naturally occurring human peptides.
We extracted the natural peptide. We didn't make any predictions. We purified the real peptide from the body of humans, and purified it and put it on the pancreas, and it worked. And it turned out to be the same peptide, of course.
One approach synthesized by hypothesis. The other identified and sequenced naturally occurring peptides. What is hard to believe is that they discovered GLP-1 at nearly the same time. Each scientist's research validated the others, confirming over and again GLP-1 7-37's great potential for diabetes treatment. At that time, diabetes treatments mainly involved insulin injections or oral small molecule drugs to lower blood sugar. GLP-1, by contrast, regulates blood sugar through the natural mechanisms of the body, thereby minimizing the side effects.
An improvement over giving insulin injections to patients with diabetes, because insulin can cause hypoglycemia, that's the lowering of the blood sugar, and actually cause loss of consciousness, and it can be lethal. Whereas the GLP-1 that stimulates the body, the pancreas, to make its own insulin, shuts itself off when the blood sugar gets low. So that suggests it could be a better treatment for diabetes, type 2 diabetes.
Habener's team launched human trials in 1989 for subjects with type 2 diabetes. They instructed subjects to eat within 30 minutes after receiving GLP-1 injection. The trial showed that GLP-1 lowered blood sugar in the diabetic patients. it also revealed something intriguing.
Stimulate, it's part of the experiment. And they said, I'd like to cooperate, but I can't. I'm full. So it made it, he got full fast and couldn't eat the full meal. So it inhibited eating. And if you think about it, it's a no-brainer. If you don't eat, you're gonna lose weight.
The team happened to observe GLP-1's weight loss effects, but their important discovery was not immediately recognized by the scientific community. They couldn't find a journal to publish it, mainly because the sample size was too small. This left the scientific community skeptical. While Holst had proved GLP-1 could stimulate insulin secretion, his work gained scant attention. It was only when GLP-1 was found to have other functions, like suppressing glucagon secretion, that it began to get noticed. Then in 1993, he ran a larger-scale human trial.
Big, important study in '93 where we actually infused GLP-1 to people with diabetes, Michael Nauk and us. And that's a wonderful paper. That's the real important thing.
Research continued, but the major obstacle to clinical use was GLP-1's very short half-life. Getting from discovery of GLP-1 7 to 37 to the development of drugs took several challenging decades. In a surprising turn, the first real GLP-1 drug involved a lizard. In 1992, scientists isolated a hormone similar to GLP-1 from the poisonous saliva of the American Gila monster. With its unique structural differences, the hormone can resist enzyme DPP-4, which inactivates GLP-1, allowing it to last in the body for hours and regulate blood sugar over a longer time. This was later developed into exenatide, the first GLP-1 receptor agonist, which gained approval in 2005.
Is because it showed that peptides can be effective medicines. Before that, there was reluctance in the pharmaceutical industry because they thought you need to come up with small molecules because they're organic, they're cheaper to produce, that people will be more likely to take it. But having exenatide at the market, it really changed the whole outlook of developing 7 to 37 medicines.
Exenatide's success opened the path for multiple GLP-1 agonists, including the widely known and widely used drug semaglutide. While altering GLP-1's structure did extend its half-life, Holst's team found yet another solution, targeting DPP-4 directly.
We showed that if you could inhibit the enzyme, the body's own GLP-1 would survive better. And sure enough, this happened and became to the development of the DPP-4 inhibitors, which are, as you know, being sold all over the world, have been for the last 20 years. Really, really an amazing. I'm very proud of that story because it was so beautiful.
The oral drug Januvia, the first DPP-4 inhibitor, was approved for market in 2006. It is still very effective in treating mild diabetes. Due to Holst's long-term cooperation with Novo Nordisk, his relentless push for GLP-1 finally caught the attention of pharma insiders. Professor Catherine Orskov, who joined Holst's lab as a student and now serves as his director, recalls that time vividly.
Also started collaborating with a company called Novo Nordisk and also talking to other companies, trying to convince people who would actually be able to use this to the benefit of mankind.
Holst made major key contributions to the development process. But for him, it was never about money.
Pushing for the success, for instance, of the drug at Novo Nordisk, I mean, sort of keeping saying it's important for this reason, for that reason, but without having a money involvement himself. So it's not like he's not become rich because Novo Nordisk did it.
What really made GLP-1 a weight loss drug was a study that increased its dosage.
That's how it began. And it was because they had this, some people out there had this crazy idea that perhaps if you turn up the dose, then you can get more. We had shown already in 2001 that the effect on food intake was dose dependent. The more you could give, the more you could inhibit food intake. But on the other hand, we also had the side effect, we had the vomiting and the nausea. So it was always a balance between the two.
Simply increasing the dosage of the diabetes medications made them effective for weight loss. This key discovery led to Saxenda, the first GLP-1 weight loss drug, which was approved in 2014. Novo Nordisk ran further trials with a raised semaglutide dosage, which led to an even more effective drug.
And that caused the basis for the registration trials for Wegovy, the STEP trials. And in the STEP trials, they found a weight loss by 2.4 milligram of up to 18%. That's when the craze began. That's when the real craze began.
From the turn of the century, the number of obese people has surged in modern society due to environmental factors like diet and lifestyle. It is a massive challenge to global public health. Diabetes and obesity are closely linked. Diabetes sufferers put on weight easily, and obesity can cause diabetes. It is a vicious cycle.
Hundreds of millions, a billion people with obesity, and hundreds of millions of people with type 2 diabetes. So these are very common. They're common disorders, obviously, in the United States. They're very common in Taiwan and very common around the world. And so these disorders still remain among our biggest health challenge because it affects the health of so many people, and they keep rising. Despite everything we do, they keep rising. Obesity is a cause of insulin resistance, and insulin resistance is the major driver for type 2 diabetes. But insulin resistance can be caused by many factors besides obesity. We have a certain genetic quality. If you are in a family that has type 2 diabetes, you probably have already an elevated genetic level of insulin resistance. What happens when you then become obese on top of that is it makes it more resistant. And then when you're more resistant, your pancreas really can't keep up with the demand to make more and more insulin. So you will, in some cases, develop diabetes.
GLP-1 receptor agonist drugs are able to both treat diabetes and aid in significant weight loss, transforming care for both conditions.
And it wasn't until recently that we have medications that not only treat the blood sugar, but also are disease modifying, that have an effect on how the disease progresses. And because of the many actions of GLP-1 receptor agonists, not only are you addressing the glucose, but you're also modulating weight loss, energy utilization, appetite, gastric emptying, and a number of different things. So for the first time, we have something that has multiple effects that modify the disease process.
These drugs are also beneficial for those with other complications.
A person doesn't really care so much about the sugar being high. You care about having a heart attack, having a stroke, losing your leg because of an amputation or kidney function. And for the first time now we have a class of drugs that not only affect the sugar, but also all the downstream complications. So very incredibly exciting.
GLP-1 drugs have become a worldwide phenomenon. They had a market value of $37 billion in 2023 and are projected to climb to $149.2 billion by 2030, with demand outstripping supply.
There's definitely a shortage of the drug because the demand is so high. And the major pharmaceutical companies that make the GLP-1 receptor agonists, Novo Nordisk and Eli Lilly, make the two major products, but there are other companies now coming along also trying to make them. They literally can't make enough.
Due to strict regulations, many might be surprised to learn that in Denmark, home to Novo Nordisk, GLP-1 weight loss drugs are hard to obtain.
Of course, if you have diabetes, yes, you can have access and the state will pay it for you. If you are very obese, we have people who can, for instance, they're simply too obese even to get the obesity operation. And their joints ache and they cannot walk and they cannot work. So you have to be quite obese in order to get it in Denmark.
High costs in the US have caused a crowding out effect.
And sometimes the people who have the higher medical need have less financial means to pay. The people who want to look better have more financial means to pay. And how do we balance that? These are tricky social issues, I would say.
Recently, big pharma companies have been vying to develop the next generation of drugs with greater weight loss effects. A research team at Eli Lilly combined GLP-1 and GIP into tirzepatide, which is a groundbreaking dual receptor agonist. It doesn't just treat diabetes. Its impressive weight loss outcomes beat all prior drugs and gained FDA approval in the US in 2022 and 2023. GIP was once thought to have no function, so this discovery came as a shock to the scientific community. This also sparked Holst's curiosity to find out why. He then published some remarkable findings with Lilly's scientists.
And we've actually published together with the people from Lilly why this happens. It happens because the interaction with the GLP-1 receptor of this compound is stronger than normal. The GIP doesn't do anything as usual, but the GLP-1 part is stronger. So it's a super GLP-1 agonist, and the addition of GIP doesn't do anything. That's the secret.
In the latest developments, a glucagon agonist has been added for a triple receptor agonist. Although glucagon aids in treating fatty liver as well, it may actually have more side effects.
So you may create muscular wasting with that. And you also interfere with the heart, so you have a further increase in heart rate, and you also produce more side effects in terms of nausea. So there are some problems in adding in a glucagon moiety in a triple agonist. And let's see how that pans out. Let's see.
However, this new path makes Mojsov hopeful for the future.
Open a whole new frontier, which shows that you can design even more effective medicines for treatment of obesity by combining pancreatic and intestinal peptides. And so I am really very optimistic that we will have even more effective medicine to treat obesity.
Through extensive research, scientists have ample evidence that GLP-1 can aid in weight loss. It does more than reduce gastric acid and slow stomach emptying. It also lessens the appetite via GLP-1 receptors in the brain. And studies suggest this may be for more than just food. It shows promise in other areas as well.
So aversive means you get turned off. Some sensation is kind of turned off. People who take GLP-1 apparently, lose less, they get less interested in drinking or less interested in smoking or so they lose certain behaviors in a general way, and we don't know quite how that works.
Many teams are now considering its use for addiction. In spring 2024, an encouraging clinical report was published on a trial with 80,000 participants.
Cannabis abuse was also addressed, and with a fantastic cure rate of 50%, were able to get out of it while they were in therapy.
And using GLP-1 drugs in treatment of Alzheimer's disease is being investigated in pharma-run clinical trials. It has been noted that GLP-1 drugs may potentially prevent colorectal cancer, and they show significant potential in treating cardiovascular and inflammatory diseases.
Like rheumatoid arthritis has just been reported, and psoriasis, inflammatory skin disease. And then when you think about it, atherosclerosis, hardening of the arteries, which causes strokes and heart attacks, is inflammatory disease of the intima of our arteries.
Since GLP-1 receptors are present in several organs, the drugs may be applicable to a wide range of diseases.
No, I would think it's very exciting, and it's not surprising because there's GLP-1 receptors all over the body, and so it's not surprising that it has effects on other parts of the body. And it's exciting that you can investigate, but it should be done in a rigorous fashion. There should be the studies that are done that they prove that the drug has the beneficial effects.
This potential brings great optimism to the medical community, with visions of even greater developments ahead.
I mean, I think in terms of the next step, what you would like to see is something that causes a permanent, in other words, that, right now many of the effects, to be sustained, you need to continue taking the therapy. So you're not really curing the disease. You're not really curing, you're simply treating it in a way that modifies the disease process, so that's great, but you need to take it for a lifetime to be able to see the effects. So ideally, what the research is moving should be one, not only what other effects does it have, but also can we make these effects permanent so that you're not required to take the medication forever.
People receiving treatments for diabetes or weight loss must use these drugs over the long term. Scientists stress that we can't depend on medication alone.
As you lose weight, you also lose lean body mass. And this could be a problem. Once you're there, after the final weight loss, all your muscles have gone. Too bad. So that's why you must increase your physical activity. You must maintain your lean body mass. And you can do it. We've shown this. We had a beautiful paper in the New England Journal of Medicine just last year on this, on a combination of physical exertion and liraglutide in this case.
Holst remains highly active and is engaged in several research projects. A born leader, he also likes sharing insights.
He's been very good at, I think, making sure that they got credit, because that's also, if you're a young researcher, it's very important because he doesn't need, he has a lot of publications. So I think he's been pretty good at sort of giving people credit and putting them on papers.
What's most admirable is despite his fame, he remains true to his character.
He's always been a person, which I appreciate, who always said yes also to teaching, for instance. I mean, he would also say, no, I'm such an important researcher, I cannot do teaching, I don't want to do the students. So he's always been like going first, like also teaching our medical students, also basic physiology, helping with exams. I mean, he's been a totally normal colleague who has behaved like a colleague, and I think that's very important.
Habener, who spent many years at Mass General Hospital, is now retired. Professor Ronald Kahn, who's known him for decades, says Habener's success comes from his love of knowledge.
His own intellectual curiosity and persistence, which was exemplified by this identifying of this hormone structure and then seeing the other hormones in it, is he wasn't really looking for this hormone. He was trying to understand the hormone glucagon. This kind of research would not occur if you were totally focused on the one discovery. You have to have an open mind.
What's more, Habener is concerned not just with research, but also on clinical applications.
He is that kind of scientist, principled, but at the same time with a clinical perspective, and then also very generous. So he, in the recent years, has been able to support some of the educational initiatives in our division, and also has trained an amazing number of people.
Since the discovery of GLP-1, Habener and Holst have each won many awards, while Mojsov has been passed over, a situation she had trouble understanding. In the end, she took action for due recognition. She had kept abundant records, each page proof of her tireless and arduous research efforts, which she presented as her evidence. Science magazines also reported on her repeated lack of recognition, which altogether helped her secure the recognition she deserved.
So I believe that my efforts to have my work recognized really can help other investigators who feel that their work has been ignored to stand up for themselves.
Rockefeller University, where she teaches, champions women scientists. Its Women in Science initiative has been in operation for over 2 decades.
Actually, I donated the financial part of the prize to the Women in Science initiative. I think it's very helpful, and there's now graduate students, Women in Science, and I think it's very nice that you have this group which is supportive of each other.
GLP-1's story is taking off, with new directions for treatment coming one after another. In December 2024, the FDA approved tirzepatide, a weight loss drug, for treating obstructive sleep apnea. It is the first prescription medication approved for this common sleep disorder. Many of the millions of Americans with sleep apnea are also obese. These developments were entirely outside the expectations of the 3 scientists. When a journalist once asked Habener to describe his feelings in one word,
I said, wow, in one word. It gives really a good feeling to know that it's, the the research that I was involved in has helped the health conditions of so many people throughout the world.
As for Holst, he remains dedicated to achieving his ultimate goal, a cure for diabetes. In a way, he is returning to his passion.
What we have been working with a lot is bariatric surgery. And that's, that's the model of all this. Because here you have weight losses between 25 and 35%. And if you have that, then your diabetes disappears and everything is good. It's really, so this tells you what you can do, because how does this happen? The bariatric surgery, weight loss, and the cure of diabetes.
Mojsov believes that the story of GLP-1 is a perfect example of biotech and medicine working together. The process from studying biology to developing drugs must return to basic research.
And so now we are going from the clinic, we have to go back to the bench. So it's kind of a full circle. You go from the bench to the clinic, and now from the clinic to the bench. And I think that's what's so exciting.
The three scientists were overjoyed to receive the Tang Prize, and they even donated precious mementos to the foundation. Habener donated his Howard Hughes Medical Institute certificate, which recognized his decades of contributions.
I've had about, I guess, over 150 students and postdocs and visiting scientists and students working in my laboratory over the 50 years or so that I've been at the Mass General working. I enjoy imagination. It's like a puzzle.
As for Mojsov, she chose the most important page from her GLP-1 experiment notebook for the Tang Prizes collection.
This is very important when you are trying to discover a new peptide. So my first date when I started this was September 13th, 83. So you can see the way this is arranged. So these are various calculations.
And Holst decided on a precious photo from his many experiments to give to the foundation. Many of his key studies were run using pigs.
In Stockholm, when they were isolating all these peptides, they came from pigs. And there are some species differences. So, to have a relevant physiological model for all of these peptides, a pig would be a natural thing.
One century-old hypothesis has intrigued countless scientists who have each set out to solve the mystery. As they sought to understand incretins, they uncovered secrets hidden within the human organs through cross-species homology and developed truly groundbreaking drugs with the discovery of differences among species. These three scientists, in the pursuit of knowledge and with a real passion for treatment, kept their eyes on the goal, worked alongside industry and academia, and turned GLP-1 into a medical breakthrough story. The future of GLP-1 based therapeutics is bright, and the biomedical contributions of these three scientists are truly far-reaching.